Very good afternoon, dear students. We have discussed about repetitive DNA sequences and non-repetitive DNA sequences. So the next discussion is on the subtopic chromosomal rearrangement or chromosome rearrangement. So let's begin with this topic. As we think of the term rearrangement, the other meaning of it is shuffling or reshuffling. The definition of this terminology is chromosome may become rearranged internally, it may reshuffle internally, or it may become joined to another chromosome. So there are two ways of rearrangement of chromosomes. One is the rearrangement of chromosome inside the same chromosome, and one more is the rearrangement of chromosomes by exchange of chromosomal segments. So in either case, it is called a rearrangement of chromosomes because it is going to alter the structure of original chromosome, and that's the reason we are calling it as rearrangement or reshuffling of chromosomal structure. So to understand rearrangement, I have taken an example of two different species of same genus, Drosophila melanogaster and Drosophila virilis. If you see in this slide, both the images of Drosophila melanogaster and virilis look almost same morphologically. But when we consider the chromosomes present in both these species, of course, the chromosome number will not change, but the arrangement of chromosome is different. So that's what is depicted in this slide. In Drosophila melanogaster, as well as in virilis, four pairs of chromosomes are found, including one pair of sex chromosome. One pair of sex chromosome and three pairs of autosomes. In Drosophila melanogaster, two pairs of large metacentric autosomes are present, where centromere is present in the exact middle of the chromosomes. Whereas in case of Drosophila virilis, four pairs of acrocentric chromosomes are present. They are called acrocentric because centromeres are found near one end of the chromosome. So that is the difference between Drosophila melanogaster and virilis. Even though they have same number of chromosomes, the arrangement of chromosomes is different, like two metacentric chromosomes are present in melanogaster and four pairs of acrocentric chromosomes are present in Drosophila virilis. Then if we try to think why such variation is required, does it really require variation? The answer is yes. Even in the same genus, different species can have different chromosomal arrangements. These differences in the arrangement of chromosomes tell us that during the course of evolution, segments of genomes are rearranged frequently. The observation that chromosome rearrangements can be found as variants within a single species suggests that genome is continuously being reshaped. The meaning of this sentence is even in the same species, if I try to analyze the chromosomal arrangement in my cells and in some other person's cell, it will not be same. It will be having little differences. So that tells us the idea about the reshaping of genome is a continuous process. So continuously our chromosomes are being reshaped. These arrangements may change the position of a segment within the chromosome. So what for these arrangements are done? These arrangements are done to change the position of the segments within the chromosome, or they may bring together segments from different chromosomes. That means two different chromosomes may change their segments, or a single chromosome may. So these arrangements are of two types. One is inversion, which involve a switch in the orientation of segment within a chromosome, within the same chromosome. A particular part of a chromosome is flipped, reverted, and again inserted back in the same chromosome. That is called as inversion. And there is one more possibility where the segments of two different chromosomes may get interchanged, and such rearrangements are called translocations. So we'll see what is inversion and how it occurs, and what is translocation, and what is a process involved in translocation, one by one. Inversion occurs when a chromosome segment breaks. A small portion of chromosome breaks, or it detaches from the chromosome and flipped around a 180 degree. So it will take a turn and then again reattach to the original position. As a result, the order of segments gene is reversed, and this process is called as inversion. And this inversion is mechanically induced by the shearing of chromosomes, or one more factor which is responsible for inversion is transposable elements. As we know, transposable elements jump from one region to other region. While jumping from one region to the other region, when they are cut or copied from one region, they will be flipped around a 180 degree and then they will be inserted on the other region, changing the structure of the chromosomes. One more factor which is responsible for inversion is continuous exposure of chromosomes to the X-ray or X irradiations. It may break the chromosome and the broken piece will be flipped and then it will be reattached to the original chromosomal segment, making the inversions. This diagram is going to tell you about how inversions occur and what are the two types of inversions. So as you can see in the first diagram A, it is called as pericentric inversion. The inversion which involves centromere is called as pericentric inversion. So as you can see, the genes present on the chromosome are labeled as A, B, C, D, E, F, G in the first diagram. And there is a cut between D and E as well as G and H. So this cut portion will be flipped and again rejoined at the same position. And now the rearranged chromosome will be read as A, B, C, D, G, F, E, H. So as it involves the centromere region, it is called as pericentric inversion. So this is first kind of inversion, and there is one more type of inversion which doesn't involve centromere, which excludes centromere. Like if the cut is made between BC and EF, the CDE portion will be flipped at 180 degree and again inserted back to the same position, and the sequence is now read as ABEDCFGH. So such type of inversions are called paracentric inversion, which doesn't involve centromere. So usually pericentric inversions are easy to detect, whereas paracentric inversions are very tough to detect, because in case of pericentric inversion, the length of chromosomal arm may change. Listen here carefully. In case of pericentric inversion, the length of chromosomal arm is getting changed in this diagram. Whereas in case of paracentric inversion, after inversion also there is no change in the length of chromosomal arm. So that's why it is quite difficult to detect paracentric inversions, whereas it's quite easier to detect pericentric inversions. So when this inverted chromosomes and non-inverted normal chromosome, they want to pair with each other during meiosis. As we know, during meiosis 1, there is a pairing of homologous chromosomes, and when they try to pair, the original normal chromosome will form a loop-like structure which helps in bringing the actual complementary segments opposite to each other. So normal chromosome will form a one turn, or it is called as loop, to help in bringing the complementary genes opposite to each other. So that's what is occurring during the pairing of inverted chromosome with that of normal chromosome. So always this inversion, when we are speaking about inversion, we are speaking of homologous chromosome. We are not speaking of non-homologous chromosomes. So in this picture, you can clearly see what is translocation. There are two pairs of homologous chromosome. One is indicated in blue color and one is indicated in green color. So there is a exchange between non-homologous non-homologous chromosomes. Green segment is given to the blue chromosome and blue segment is given to the green chromosome. Such type of rearrangement is called as translocation. And translocations are quite common during crossing over. The pairing of chromosome in translocation heterozygote occurs in this way as it is depicted in the diagram. A small portion of one chromosomal segment will be given to the other and a small portion from that second chromosome will be taken back on the third chromosome. There are three different ways of disjunction after exchange of chromosomal segments. These chromosomes are segregated in three different ways. One is adjacent disjunction 1, adjacent disjunction type 2, and third is alternative disjunction. In all the three cases, you can find second and third chromosomes are involved in the rearrangement, but these different types will tell us to which side the chromosomes are pulled apart. Pulled. In first case, centromeres 1 and 3 will go to one end, whereas 2 and 4 will go to other end. In the second case, 1 and 2 will go to one end and 4, 3 will go to opposite end. Whereas in case of alternate disjunction, they will be flipped. 2 and 3 will go to one end and 1 and 4 will go to other end. That's why it is called as alternative disjunction. So totally either it is inversion or it is translocation. They are going to change the structure of the chromosomes, and they are very essential for evolution. Thank you.